Welcome back, friends. Welcome to another video tutorial from Shomu's Biology. And in this video tutorial, we'll be talking about NMR spectroscopy. Now, this is going to be an overview and a basic idea about NMR spectroscopy and what is NMR spectroscopy in the basic scale. The full form of NMR spectroscopy is nuclear magnetic resonance spectroscopy. Now, if we carefully break this name down into fragments, it's going to give you an idea about what we are talking about here. Nuclear magnetic resonance. There are some lot of terms over there. Nuclear, why? Because NMR spectroscopy is a technique with which we identify the presence of different chemical and physical properties of organic or molecules, or inorganic molecules that we deal with. Let's say protein. You want to find the structure of an unknown protein or structure of any other compound, chemical compound. In that case, that compound is associated with so many different atoms, and they are arranged in different ways. Now, they have different physical properties as well as chemical properties. That is mainly due to the presence of different atoms present there. Now, the thing is, with the help of NMR spectroscopy, we can study those chemical and physical properties of all those molecules or chemical molecules that we are talking about. Okay. Now, there are different ways of identifying the unknown structure of those chemical compounds. One of those is NMR. There is IR spectroscopy. There is mass spectroscopy. There is X-ray crystallography that helps us to identify the unknown structure of molecules, even big molecules like proteins or deoxynucleotide acid or nucleic acid. So, this is the idea of NMR spectroscopy in this video. The first thing I want to talk about is that NMR spectroscopy exploit the idea of spinning and the feature of electron as a magnet. So, what we mean? Normally what we know, as it says nuclear magnetic resonance, that means all of these atoms, they have a nucleus. We know that atom contains a nucleus, and the nucleus is surrounded by electrons. That's the idea. For understanding NMR, we'll start to talk about the simpler form of atoms and molecules, then we'll go to the complex form. For example, say if we begin with hydrogen, hydrogen is a very simple, it contains a very simple structure because hydrogen have only one proton and one electron. The proton is in the middle in the nucleus, and the electron is circulating this proton. That's the idea. That's the structure of the hydrogen. Now, here in this process of NMR spectroscopy, we will see this proton playing vital role. What kind of role? That this proton can spin. It can spin. Due to the spinning property of the proton, it can behave like a magnet. Okay? And this is not only true for hydrogen. This is also true for many other type of atoms. For example, nitrogen, carbon, chlorine, all these things, they have the same property. They have the proton, and that can spin. The reason for the spinning, that's the reason that this proton are spinning. Due to the spin, they get the magnetic property. Now, for the simplicity's sake, we only take hydrogen and explain about hydrogen in the process of NMR spectroscopy. Now, as I told you, like the hydrogen can spin, so I told you like it can act as a magnet. So, let's assume this hydrogen acting as a magnet, and if it's a magnet, it obviously have a north pole and a south pole like every magnet. So, the north, we put an arrow. How we designate a magnet? We put an arrow to the north, just like a vector representation, because that is the direction where the magnet always pointed to. So, this is the hydrogen behaving like a magnet. Now, one thing you should understand, once it is a magnet, let's put another magnet close to this hydrogen. That could be another hydrogen atom. So, if you put that hydrogen atom very close to this one, let's say we bring another hydrogen with north pole towards the north of this hydrogen. So, this north will repel each other out. So, it will try to rotate the other one in the opposite direction. It will allow this second atom to spin so that the south pole gets near to the north. So, they will have a rearrangement. and this is true for any magnet that we know of. And this rearrangement is really important, because after the rearrangement, whatever structure they acquire, that is known as the stable state or least energy state. That is the most economic state this atom can be. So they want to be in those stable state. Now imagine the situation, this hydrogen is present, and if we put some kind of magnetic field means we are providing a direction of north with the help of a machine that will also do the same type of effect, right? So let me draw that same thing here. Let's say here in the machine we are providing this direction of the arrow is the field towards the north. So this is the north field, and this machine is known as the NMR machine. So the job of NMR machine is to create a magnetic field. As we are creating the magnetic field, what we know is that in whatever direction that hydrogen was, let's say this is the north, this is the south, in whatever direction it was before, now it will be shifted to only one direction, that is this one. The north will be same as the north of the magnetic field, the south will be same as the south of the magnetic field. So they will be rearranged, because this is the steady state, this is the most stable state. Okay. So let's say normally the molecules generally don't present like this, they present in any orientation, but the atoms present in any orientation, the hydrogen present in any orientation. But whenever we apply this magnetic field, they will have a quick spin so that they match the spin of the magnetic field. That spin is known as the alpha spin. Remember, this is known as the alpha spin. Once this spin is done, they will be rearranged to the direction of magnetic field. So this is a general idea. So what we have learned till now, we know that this atom hydrogen can behave like a magnet. Whenever we can apply some kind of magnetic field, it can have a spin to match that field. That's true. And in the NMR machine we do exactly that thing. We apply some magnetic field so that according to the field, all the molecules and the atoms present in the molecules rearrange according to that magnetic field. Okay, so far so good. Now the second part of this understanding is how exactly it is going to help us understanding the structure of molecule. How do you know that? Now this NMR spectroscopy is very important to know the presence of different groups and presence of different atoms with one another. So it's very important in the spatial arrangement of those atoms with respect to one another. Example for that, it could be chlorine, carbon, hydrogen, for example, like that, or it could be like hydrogen, carbon, hydrogen attached with chlorine, like that. So if you look at these two scenarios here, chlorine is present, one carbon, two hydrogens are present. The same thing may happen like that, but the organization and the arrangement of chlorine is different in these two cases. Normally IR spectroscopy or mass spec, those are not very good to distinguish between these two molecules, because they have the same number of carbon, same number of chlorine, same number of hydrogen. So they might end up with telling these two molecules are the same, but it can be different, and this difference can be identified only with NMR, because NMR can give you the idea about how other atoms are placed with respect to their positions and attachment with the nearby atom. Okay, so now if we look here, the whole idea about the spin. So we talked about till now that if you put the magnetic field, this proton have a spin, this hydrogen have a spin. Now they match that, so it's called the alpha spin. So it's easy to get the alpha spin. Now we take it a little far. Now what we will do, Now let's say we want to apply some energy so that we can turn this hydrogen opposite direction, 180-degree opposite direction. That means we want this north to go towards the south. We want it to be completely reversed. So definitely we'll need a lot of energy to do that. So what we do, we put a electromagnetic wave, another kind of electromagnetic wave. It's a normal light. If you look at the normal light frequency, it's a normal wave. That wave or that light source can provide the sufficient energy required for reverse spinning this hydrogen atom. So it can reverse spin. When it spins back to the opposite orientation, let me draw the opposite orientation here as well. Let's say these are the directionality of the magnetic field. Let's say this is the one, and it is organized in the opposite direction. So you see the arrow is completely 180-degree flipped. Now this spin is known as beta spin. We know alpha spin, which is the same directionality with the magnetic field, the organization, while beta spin is the opposite direction of that magnet to the magnetic field. These are the two types of spin that we know of. Now the thing is, this situation is only possible if we put some extra energy with the help of some electromagnetic radiation. We put the wave, the light, that can provide sufficient energy to do that. Okay, so once they reach this alpha to beta, we need some energy to do. But now remember one thing, this is the most important point about the NMR spectroscopy. As we are talking about one type of magnet, that is hydrogen, for example. Now the hydrogen, it could be different other atoms present nearby hydrogen, for example nitrogen, carbon, oxygen, different molecules can be present, different atoms can be present. Now this is very important, how this hydrogen is interacting and how this hydrogen is placed near all those different atoms. That's what makes this process a technique to specify and identify the chemical and physical properties of the molecules. For example, here you say that this hydrogen is present very near to carbon here and the other one is far downside. But here you see the hydrogen are present very close proximity to the carbon, in the same close proximity to the carbon. So it depends how exactly hydrogen is arranged. That is a proton working as a proton here only, because it contains only one proton there. So the arrangement is very, very important in this case of NMR spectroscopy. Now I'll be giving you another idea about how the presence of other molecules near hydrogen, other atoms, I'm very sorry, it's not molecule, other atoms near hydrogen changes its behavior. Let's look at that. Now let's say hydrogen is present, carbon is also surrounding that hydrogen. What does that mean? Hydrogen have only one electron, and we are not going to talk about that much because it's only one electron, the effect is not that high. But it has the proton, only one single proton, and we are talking about the proton because it is the proton's feature to give us the magnetic field or give or behave like a magnet. Now this nearby carbon molecules contains a lot of electron, right? They contains a lot of electron. So the idea is these protons sometime, let's say this is the proton acting as a magnet, this proton is surrounded by a lot of electrons. So this magnet is masked by a lot of electrons. We call it as a magnetic shielding or call it a diamagnetic shielding. This diamagnetic shielding is actually preventing this magnet feature of the hydrogen. Okay. So for different molecules this magnetic shielding could be different, right? So let's take a molecule where we have only one hydrogen, and let's take another molecule where we have hydrogen attached with carbon, two different carbons for example nearby, right? Or let's say take one molecule where we have one carbon and hydrogen, and another molecule where we have two carbons attached to the same hydrogen. So as there are two carbons present, this second one will be more shielded. The first one have only one carbon, so it will be less shielded. So what does this shielding affect the whole idea about spinning? Let's look at it. The idea about the spinning and shielding, again I have to draw that. Let's say this is the same scenario that we are talking about. This is the direction of the magnetic field in the NMR machine, and this is our proton. This is a hydrogen that we are talking about. It is in the alpha spin mode. Now I told you that we want sometime this magnet, this hydrogen, to be completely reversed spin to the beta state. So this reversing will be different for different type of molecules depending upon the presence of those electrons. If there are no electrons present surrounding this hydrogen, the magnetic effect will be very high in that sense. So what we need more of this energy to shift it to the beta state. Okay, but let's say if this same scenario and it is filled with electrons, surrounding with electrons, these electrons are masking the effect of the magnetic field. So the effect of magnetic field is not that high in this hydrogen atom. So we need less energy as electromagnetic radiation, electromagnetic wave, to put it to the beta state or beta spin state. Got the idea? So if it is shielded, we need less energy to spin it back to the beta state. If it is not shielded, we need more energy to put it back to the beta spin state. This is the most important point that help us to find out the structure and the arrangement of different protons, and arrangement of different atoms in the molecule. Okay, how? Let's look at it. This is filled, so I have to erase this, and then I'll explain how exactly we'll determine the structure of a molecule based on the idea of this shielding and less shielding. Okay, it's simply because for different molecules we have a different arrangement of these atoms, different bonding of these atoms, and due to the presence of different atoms nearby, this shielding and non-shielding will take place, and that will help us to find out the exact structure. Okay, so let's look at that in a moment. Is this idea of spinning the magnet back 180 degree to the beta spin state, and that thing is known as the resonance. Resonance, why? Because if you are talking about consecutive atoms that are linked with a single atom, if you're talking about that, resonance means we want to put all those atoms or all those hydrogens that we're dealing with, which are behaving like a magnet, to the same orientation with the magnetic field. That will be the opposite of that magnetic field. Okay, so at a time, if you're dealing with a molecule structure, we definitely want all the hydrogen that is present in that atom, that is present in that molecule, that should be spinned in the same direction, same orientation, and that same orientation of the spin is going to give us a data that is going to help us to understand about the structure of that molecule. How? Because as we are telling all those hydrogen to get a same type of arrangement which is against the magnetic field, this is known as a resonance, because resonance means all those other atoms are doing the same thing at the same time. That is called the resonance here. So the idea is very simple. Earlier the hydrogen was present according to the magnetic field, the direction to the magnetic field. Now we apply some energy here. The energy is applied with the radio electromagnetic wave or radio wave, you can tell, that will shift this orientation of this hydrogen to the opposite side. So we have a beta spin here. Now for orienting into the beta spin, as we saw, if it is shielded by electrons, we need less energy. So if it's shielded, we need less energy. If it is de-shielded, we need more energy. So what we do, if we do the graph, let me do a structure or a graph. If we do a graph, in the x-axis we have the energy. Okay, so what we will find here is that they will be like at the from the beginning, there will be peak that we observe, peak that we can see based on the same hydrogen that we are dealing with. The length will be the same, let's say the height will be the same. It's not that blunt, it is okay. So this kind of peak that we'll observe. Now for example, say there is a shielded and de-shielded, two types of magnetic situation we are talking about, two types of hydrogen we are talking about. For the shielded one, remember, we need less energy. So the energy that will for the shielded, which will be less, which will be de-shielded, that will be here. Okay, shielded and de-shielded. We have this kind of graph for that. So this kind of graph is going to tell us about how exactly it is organized and how exactly we are talking about. Now think about the same graph if we are dealing with a molecule which have multiple hydrogens attached with different molecules there, attached different atoms. We have a different kind of graph, and the pattern of the graph is going to tell us about what kind of atoms are organized and arranged with each other, where exactly the hydrogen is present, how many hydrogen is present or not. Because here hydrogen is very important, and the presence of hydrogen and how hydrogen is linked with other atoms plays the most important role about how it is going to determine the whole process and the structure of that chemical compound. Okay. So now let's see one chemical compound and how exactly NMR determines its structure. Okay, let's say the compound is ClCH2CH3, for example. Now in this compound you will see there are 2 hydrogen here and 3 hydrogen here. So there is a lot of hydrogen present there. And actually if we are talking about the organic chemistry, and most of these organic compounds, they are hydrocarbon. So they are filled with carbon and hydrogen. So hydrogen is present in all those molecules, and those hydrogen will be very beneficial for us to find out the exact arrangement of those atoms that forms that chemical compound. Okay. So now let's look at, in this case, if you think of this hydrogen in the middle, let's take one hydrogen that is present here. Okay. So the hydrogen that is present and attached to the carbon, so the nearest attachment or nearest atom for the hydrogen here is this carbon, right? That means this hydrogen, let's say this is the hydrogen, it will try to behave like a magnet. It will be arranged according to the magnetic field, let's say like that, but there is this electrons coming from the carbon, the nearby carbon. So there will be electrons from the nearby carbon that will not allow the effect of the magnetic field that we apply in the NMR machine to this molecule. As a result, this hydrogen will not properly align with, this magnetic field, because it is shielded by the electrons coming from the nearby carbon. So this is a shielded situation. Now if we put the radio wave light source, that will be acting as an energy to shift it to the beta spin state, we need very less energy because it's already shielded with electrons. We need less energy to put it back to this structure, right? So they need very less energy. But now let's think of this idea a little deep. Now if you just think of this carbon and hydrogen, you may think like that. But there is a chlorine present next to the carbon. What does that mean? Chlorine has higher electronegativity. And what electronegativity does? Electronegativity means chlorine have the affinity towards electron. So it will take all those electrons towards itself from the carbon. So now the idea, electrons are surrounding this hydrogen, masking the effect of the magnetic field. But now as chlorine is present very near to the carbon, it is dragging electron towards itself due to its electronegativity. As a result of that, this hydrogen is not properly shielded as it supposed to be, right? If the chlorine is not present, then it should be shielded, and we need less energy to put it back in the beta spin state. But now as chlorine is present nearby, it is taking the electrons, so now the hydrogen is not that shielded, so we'll need more energy to deal with it. And if you take another hydrogen that is present here, that is this one, this second hydrogen here, there is no chlorine, no electronegative molecules nearby. The chlorine is present far from this hydrogen. What does that mean? Electronegativity is a very nearby process. It's a very local energy, a local process. It is not spread across the whole molecule. It's only spread across a specific region surrounding the chlorine. So this hydrogen do not have any impact of this electronegativity of the chlorine that is far apart from it. So this hydrogen will be shielded by all the electrons coming from the nearby carbon, because this hydrogen also have a nearby carbon, right? So this hydrogen will be shielded by electrons, so we need less energy to shift it back to the beta spin state. So now if we take this two as a two population, this hydrogen as A, this hydrogen as B, what we can see in the actual peak that will be detected by the detector at the end of the NMR machine. Now if we look at this detector and if you look at the monitor screen, there will be a graph, and that graph is telling us about the energy which starts from this point, and the energy is increasing in this direction towards the arrow, and we have this intensity for there. So what we will find out, that the energy required for shifting this B hydrogen is less. So the B hydrogen will be present representing the first peak. So this will be the peak for B. For A, this A hydrogen, there will be more energy required. So A will be present in the distant from the y-axis here. So this is going to tell us about where exactly the B and A is present. Now this is a very simple example to tell you what kind of situation it may arise. Now just think about the presence of all those different special atoms surrounding the hydrogen. You will find a lot of peak like that, a huge amount of peak just going up and down. It's called the peak. Okay, peak is the graph, the point that is recorded due to this resonance energy, the energy that is required. So this resonance energy peak that we'll see, it will be characteristic for different chemical compounds. For this compound, this is the character. For any other compound, the character will be different. Or let's say you take this chlorine from here simply and put it back next to this carbon, attach it to this carbon. In that case, you will have a different peak observed. So this arrangement of peak is going to be like a signature for a simple chemical compound. So once we analyze different chemical compounds with NMR spectroscopy, the presence of this hydrogen will be very important to find out the associated atomic interactions that are going to give us different peak in the graph, which is going to tell us about exactly the chemical component structure. That is the idea of NMR spectroscopy. And I hope you understand about this whole process of NMR spectroscopy in the basic level, in the overview. Okay, and NMR spectroscopy mostly determined on the nuclear magnetic resonance. That means it based on the small nucleus, that is the hydrogen atom, the nucleus of the hydrogen, which contains only one proton, and we are relying on the resonance energy that we require to put it to the beta spin state. That's the name, nuclear magnetic resonance, because all those nucleus of all those atoms like carbon, nitrogen, oxygen, hydrogen, they act as a magnet in those feature, right? So that is the idea about NMR spectroscopy, or nuclear magnetic resonance spectroscopy. Okay, if you like this video, please hit the like button, share this video with your friends, and subscribe to my channel to get more videos like that. Thank you.